Today in biology, kingdom Prokaryote Monera, you will learn about nutrition of bacteria. Image of bacteria is shown. Like other organisms, bacteria need energy for their growth, reproduction, and maintenance functions. Most bacteria are heterotrophic, unable to make their own food. That is, they can't synthesize their organic compounds food from simple inorganic substances. Again, picture of bacteria is shown. Bacteria live either as saprophytes, that is feeding on dead organic matter, or as parasites living on their hosts. Saprophytic bacteria get their food from dead organic matter. As you know, bacteria are also decomposers, so they feed on dead, decaying organic matter. Soil is full of organic compounds in the form of humus. Now, what is it? It is the organic component or part of soil formed by decomposition, breakdown of leaves, plant materials by soil microorganisms, just like bacteria. Picture is shown. What is humus? Humus is dark colored organic matter found in the soil and is made up of decayed, decomposed, dead plants and animals. Humus is the material resulting from partial, half decay, decomposition of plants and animals. Many soil inhabiting or occupying bacteria have very extensive enzyme system. This enzyme system breaks down the complex, large substances of humus into simpler, smaller compounds. That is, this decomposition is done by bacteria. Bacteria can then absorb, utilize, or use these simpler substances as a source of energy, as a source of their food. Parasitic bacteria live on their host and they're fully dependent for their nutrition on their host body. Images of parasitic bacteria on their host are shown. Why they live on host? To cause so many diseases. Some kinds of bacteria are autotrophic. Autotrophic means they are able to make their own food, having some pigments. So they can synthesize their food organic compounds which are necessary for their survival and growth from inorganic substances. Organic compounds are those in which carbon and hydrogen together are present, while inorganic compounds are those in which carbon and hydrogen do not exist together. They are able to make their own food by pigments. Pigments are the substances that absorb light energy and then convert them into food or chemical energy. Some kinds of bacteria may be separated into two groups. These are photosynthetic bacteria and chemosynthetic bacteria. It will be discussed one by one. Number one, photosynthetic bacteria. As the name suggests, that they definitely have chlorophyll pigments to trap light energy. They possess or have chlorophyll pigments which differs from the chlorophyll pigments of green plants. Unlike most green plants, which have their chlorophyll pigments in special organelles, chloroplasts, that is why they are green in color, because they are having chlorophyll. But bacterial chlorophyll is dispersed or scattered in the cytoplasm. Plants' chlorophyll is present in chloroplast organelle, while bacterial chlorophyll is present in the cytoplasm. This is the difference. During photosynthesis process, autotrophic bacteria utilize or use hydrogen sulfide H2S instead of water as hydrogen source. So they liberate sulfur instead of oxygen. Nitrifying bacteria are chemosynthetic. In leguminous plants or legume plants like pea, there are some root nodules in which bacteria live. Function or role of these bacteria is to convert atmospheric nitrogen into some nitrates which are usable by plants. Image of nitrifying bacteria is shown. Nitrifying bacteria are the microorganisms Definitely they are bacteria, and they convert soil nitrogen, ammonia into nitrates. Nitrate is the form that is usable by plants. That is, plants can use it. Next are chemosynthetic bacteria. They oxidize inorganic compounds just like ammonia, nitrate, nitrite, ferrocyanins. They trap energy released for their synthetic reactions. Simple words, chemosynthetic bacteria are a group of autotrophic bacteria. They use chemical energy to produce their own food. Examples of photosynthetic bacteria are green sulfur bacteria, purple sulfur bacteria, purple non-sulfur bacteria. Their pictures are shown, photosynthetic bacteria, so they are green in color. Photosynthetic bacteria have light-absorbing pigments, so they are able or capable of converting light energy into food energy or chemical energy. Second topic is respiration in bacteria. That is, some bacteria can grow in the presence of oxygen and some can live in the absence of oxygen. Respiration in bacteria may be aerobic, they require oxygen, or it may be anaerobic, they do not require oxygen. Aerobic bacteria can grow in the presence of oxygen. Anaerobic bacteria can grow in the absence of oxygen. Some bacteria are neither aerobic nor anaerobic. Facultative bacteria grow either in the presence or absence of oxygen. So there are different groups of bacteria on the basis of presence or absence of oxygen. Next is microaerophilic bacteria. Micro means small and aerophilic means based on the oxygen concentration. That is, they require low concentration or amount of oxygen for their growth, and so they are called as microaerophilic bacteria. Pseudomonas is an aerobic bacterium, Spirochete is an anaerobic bacterium, and Escherichia coli is a facultative anaerobic bacterium. Campylobacter is microaerophilic bacterium. Its picture is also shown. Next topic is growth and reproduction in bacteria. Bacteria are the microorganisms which rapidly increase their population. Bacterial growth refers commonly to an increase in the number of bacterial cells. That is, they grow and their cells increase in number, their population increase. Bacteria increase in number by asexual reproduction, in which single parent is involved. Binary fission also occurs in bacteria. Binary means a cell divides into two. That is, bacterial cell divides into two daughter bacterial cells which are exactly alike its parent cell. Binary fission is a method of asexual reproduction, a process in which bacterial cell divides into two bacterial daughter cells. Picture is shown that a bacterial cell splits into two cells. In binary fission, bacterial parent cell enlarges, it grows in size, its chromosome doubles or duplicates, its plasma membrane pinches inward, that is, it folds inward at the center of the cell, as you can see in the picture. That there is a bacterial cell, then its membrane or plasma membrane pinch inward, so it may divide into two cells. When nuclear material is evenly distributed, then cell wall grows inward to separate the cell into two, or to separate the cell. This sequence is repeated at intervals by which new daughter cell increases population of the cells. That is, the process is done until the cell splits into two or divides into two. Once the division is complete, then bacteria grow, develop their unique features. That is, they lack nucleus, but they have small circular chromosomes in them. Generation time. Interval of time until the completion of next division is called generation time. 4 distinct phases are in bacterial growth process: lag phase, log phase, stationary phase, death or decline phase. One by one we will discuss. Number 1, lag phase. It is a phase of no growth. That is, no growth occurs in bacteria. Bacteria just prepare themselves for cell division. Second is log phase. It is a phase of rapid growth in which bacteria divide at fast or exponential rate. Third is stationary phase. Bacterial cell death rate is just equal to bacterial rate of growth, reproduction, and multiplication. And fourth one is death or decline phase. Bacteria starts dying. Here the death rate of bacteria is more than multiplication or reproduction rate of bacteria. Bacteria lack traditional sexual reproduction and mitosis. Sexual reproduction in which both parents are involved, and mitosis, as you know, that it is a type of cell division in which parent cell divides and form two daughter cells having exact number of chromosome as their parents are having. Some bacteria transfer genetic material from one bacterium, that is donor bacterium, to a recipient bacterium during a special process called conjugation. That is, there are two bacterial cells. One is donor, which is giving its genetic material, and other one is receiving that genetic material. It is called as recipient. Some conjugating bacteria transfer their genetic material from donor to recipient using some specialized sex pili. Function of sex pili is to transfer genetic material between donor and recipient. Definition of conjugation: it is a process that allows the resulting bacteria to survive under different type of conditions. And now picture of sex pili of bacteria, you can see male genetic donor bacterium, and their genetic material is being transferred to the female recipient bacteria, and there are some structures which are transferring genetic material from one bacteria to another, and these structures are called sex pili.